team at the GDC. And then today with me, I have Dr. Zhenyu Zhang, who's the GDC Bioinformatics Manager here at the GDC. So he'll be here to answer any specific questions about the pipelines. He's developed most of the GDC pipelines that we're going to talk about here today. So in terms of the agenda for today, as a reminder, we're going to walk through the slides first and do a little bit of demo at the end. And then during the webinar, if you have any questions whatsoever, please ask them in the chat window, and then we can address them at the end of the presentation. We'll be happy to answer any questions that you have. So for today's webinar, I got it up here on the screen here. We're going to talk about the GDC harmonization process, reference genome, alignment workflow, and then the meat of the presentation is going to be actually diving into the pipelines themselves and talking about how we process the data at the GDC. So we've got the DNA-Seq pipeline, RNA-Seq, miRNA-Seq, and then copy number variation and methylation liftover pipelines. And then at the end of that, I'll just jump into like a really brief demo showing where to find the data on the GDC. And then the last slide, which may be of interest to some of you, is we have some additional pipelines that are kind of coming down the pipeline, if you will. And then we'll just briefly touch on some of those there. And then again, at the end, we'll save some time for some questions. So to get started, the GDC, again, it's a repository for clinical and biospecimen and molecular data for different cancer genomic studies. And the data that's submitted to the GDC for various projects gets processed through a set of standardized bioinformatics pipelines. So for example, the BAM and FASTQ files that are submitted to us are aligned primarily to a reference genome build, HT-38, and then processed through a set of open source bioinformatics tools, depending on whatever experimental strategies, so whole exome, whole genome, RNA-Seq, the things that I just mentioned before. And then the main benefit of doing this is such that the data can be more easily compared between different studies, and so that all the data is kind of being analyzed and produced in the same way. And the pipelines that the GDC has produces some derived harmonized data, such as, for example, gene expression count files. And the files themselves are considered controlled or open access, depending on the details of the consent for the study. And all of this data is published directly to the GDC portal. So that includes the aligned BAM files and then this derived data that I'm talking about here. And then another benefit of doing this harmonization is we index a lot of this data, such as the variant calls, so that it can be easily visualized on the data portal without a researcher having to download a BAM file and doing the analysis themselves. But that is entirely an option if you wanted to do that, since we serve up the aligned BAM files. And then the last thing on this slide that I'll mention is we've got all of the pipelines that we use very heavily documented on our documentation site. So this includes like the different tools, the version numbers of the tools, as well as the commands that were actually being used to generate the data themselves. So this slide here just is a very high overview of the current GDC pipelines that we have. So the data that you would find on the GDC right now, if you were to just log in. And it's kind of structured into a similar way as the data model, which if you tuned into other webinars, you will remember that all of the data in the GDC adheres to the standard data model that we've got here. So the red circles there are the different analyses that are being performed at various stages. And then the green circles are the actual data files that we would serve up on the GDC data portal. So you can see that this graphic starts at the aliquot level. So from that, we do our CNV pipeline on the left, and then our methylation pipeline on the right, and this is array-based data. And then going down the center of that graphic, we have read groups. We've got the sequencing reads, the alignment that I was talking about, and then from the aligned reads, we can do our DNA-Seq pipeline, microRNA, and then RNA-Seq pipelines. So for example, DNA-Seq, you can follow that down a little bit further. We do somatic variant calling, and then all the way down to aggregation of all that data into files that are more easily viewable. So yeah, that's just kind of like a high-level overview of the data we've got there. So the thing that I mentioned is the GDC generates this higher-level data that some of which gets indexed and served up on the portal. This includes DNA-Seq derived somatic mutations, RNA-Seq, and microRNA-Seq derived gene expression quantification, and then the array-based copy number and methylation data as well. And as I mentioned, all this data is on the GDC portal. So if you go to portal.gdc.cancer.gov, you'd find a lot of that data there. So the next slide talks a little bit more in detail of this harmonization process, at least the beginning of it for the next generation sequencing data. So the GDC pipelines support the realignment of DNA, RNA, and microRNA sequence data against the latest reference genome build. And this graphic kind of summarizes this pretty well. So we take, on the left-hand side, we've got the GDC object store. We take the BAM files, convert to FASTQ, do some QC on the files, and then we will the actual alignment using some of the tools that I'll mention a little bit later, and then serve up that harmonized BAM file to the GDC portal, which is controlled access. So let's actually just start doing a little deep dive into some of these pipelines we've got. So the first one is the DNA-Seq pipeline. And the main purpose of this is to identify the somatic variants within submitted whole exome and whole genome sequencing data. So the data on the portal right now will contain variants for both of those. And these are variants identified by comparing the allele frequency in a normal versus a tumor sample. And then we've got 5 different variant callers that we use to generate these somatic mutations. Those are listed there. We've got Mutect, Varscan, Somatic Sniper, Muse, and a newer one, Pindel. And then all of these, for example, one tumor and normal pair, you will expect to see one file, one VCF file. So that's that last bullet point there. So the variants themselves are described in these VCF files that you see on the portal. And then what we do is we aggregate them into a project level math file. And that graphic at the bottom kind of shows that process that it goes through. So BAM, we do variant calling, get VCFs, and then we do some mutation aggregation, and then publish a math file to the data portal. So here are the actual steps to this process. So the first step is the alignment. So the first part is we take the submitted BAM files and convert those to FASTQ, do some pre-alignment QC, and we actually have started to put some of those QC results as index files. So you can see, for example, coverage, proportion of duplicated reads, read length, base quality, that kind of stuff. That's the kind of QC we're looking at. And then we align the files using BWA, sort and mark duplicates using Picard. And then we do base quality recalibration to detect systematic errors. And the result of that is this block right here is the realigned BAM file. So this harmonized BAM file we publish to the GDC portal. So the next two steps are the actual variant calling workflow. We do somatic variant calling, and we process tumor normal pairs using the 5 different tools. And then we process through our variant callers, and as I mentioned before, the VCF file is the output of the variant calling. And we also annotate it using VEP to get some more details on that. And this next slide gives a little bit more detail on what you would expect to find in a VCF file. So for a given tumor normal pair, one VCF file will include at the top some metadata about the actual pipeline run, so the workflow case, what samples were they, and then what reference genome we use, contig, what version of VEP we use. And then the meat of it is the actual mutation calls. So it includes a column header that has information about chromosomal position, filters that were flagged, and basically that list that I've put there. And then the last thing that you would find in that info column is different fields that are annotations that come from VEP. So you see things like the gene ID, alternative names. And I didn't want to overload this slide because there's a ton of information that you'd find in the VCF file. But I will mention again that the GDC doc site will have a ton of very detailed information on what each of the columns in the VCF file means. So you can check that out. So the last part, after we've created these VCF files, we do two last steps, so aggregation and masking. So what we do is one second. So for example, for TCGA, BRCA, if we've got one VCF file there, what we do is we take all of those VCF files for that given project and aggregate them into what's called a MAF file, which is a tab delimited file that has all the mutations for an entire project. So you'd see one MAF file for TCGA, BRCA, for example. And then one of the things that I'll mention here is the mutations from some of these variant callers have the potential to be germline mutations or lower quality mutations. So there's an additional processing step that we do, which will mask out those mutations and then remove some of the lower quality mutations. And that's what this diagram is kind of showing here. So we take the variant from the protected MAF and then run it through these basically if-then statements. If it's potentially a germline mutation, get rid of it. If it has a filter, a panel of normals, then we'll include it. And then the middle bubble here it's the public somatic MAF. So basically, ultimately, for a given project for TCGA, you're going to see two different MAF files. You're going to see an open access MAF file, which is what that last one is, and then a controlled access MAF file if you want all the mutations that were identified through the variant calling algorithm. And then the benefit of having this, again, this benefit of having the open access mutation is open access MAF is we can index and visualize all of those on the GDC portal. And you will see that if you go to some of our older recordings of webinars, you can see how we can explore some of those mutations on the GDC portal. So yeah, that is the DNA-Seq pipeline. And I'm going to switch gears to RNA-Seq and microRNA-Seq. So the RNA-Seq pipeline primarily, it measures the protein coding gene expression based on the number of reads that have been aligned to each gene. And what it does is it uses a couple different tools, STAR and HT-Seq, along with, that's a typo, GenCode version 22. And what it does is basically what that schematic is showing you right here is it's taking a submitted BAM file, converting it to FASTQ if it wasn't already submitted as a FASTQ. We take BAM and FASTQ files. And then what we do is we take that FASTQ file and do splice junction detection and alignment using STAR to pass. And then the output of that are a number of different BAM files. So we have, as you can see here, the aligned transcriptomic BAM, genomic BAM, and then chimeric BAM. And what each of those means for the transcriptomic, it reports the reads aligned to the transcriptome. The chimeric one reports the reads that are aligned to different chromosomes or strands. And then the genomic BAM is what we use for the files, the output files that we have on the GDC portal. So we take that aligned genomic BAM and we use HT-Seq to quantify the expression counts. And then we also calculate a couple other things here. So we've got the raw counts and then FPKM and FPKM-UQ, which are basically normalized versions of of the count files. But what you could do is you could take the count files if you wanted to calculate something else and do that in an external analysis if you wanted to. So this is an example of one of those HT-Seq count files that I was mentioning. So on the left, you can see it's just a very simple tab delimited file. On the left, you see the gene ID. And then on the right, you see the number of reads from sequencing that overlap that specific genomic coordinate position. And what you can do with these files, so this is for one specific sample that was taken. You could do something like a gene differential expression analysis using this data. And I think this is a good time to plug. We do have a community tool that will merge all those count files or FPKM or the microRNA data that I'm going to talk about now into a matrix, which will be helpful for downstream analysis that you have. So what you can see here is on the left, you got the gene ID, and then each column represents the different sample that was taken. And this is on GitHub. You can download it. It's free. It's just a Python script that you can run. And this is kind of how people like to see the gene expression data. So the next pipeline that I'm going to mention here is the microRNA profiling pipeline. And for microRNA, we have this pipeline will quantify the microRNA and isoform expression values. And what we use for this is a modified the BC GSC pipeline that was used for TCGA. And there's a link right there if you want to go check it out and want to replicate it or run it. And then the original paper is there as well. So this schematic here is showing we take the aligned BAM file, do the quantification and normalization using that version of the pipeline, and then out of that comes two specific files for microRNA. So there's microRNA expression and then the isoform expression. And then the next slide is going to give a little more detail on each of these. So the one on the left is the quantification file. And what you can see is it's providing a list on the left of different microRNAs from MIR base, along with the read count and reads per million microRNA mapped, and a column here that describes whether it's cross-mapped or not. And then the file on the right is the quantification, or the isoforms file, and what that does is it will show you information about the coordinates in the region. What it shows you is kind of, it's the same thing, the microRNA as it corresponds in MIR base, as well as the genomic coordinates that it aligns to, and the region it constitutes in the full microRNA transcript. So you're free to download those files, and that tool that I mentioned will also do the same thing. It'll align all the files if you wanted to look at all of them on a specific project level. So keeping on this track, we have two other pipelines that I'll just briefly cover now. It's the methylation and copy number variation pipelines. So the copy number variation pipeline, what it does is it identifies the genomic regions that are repeated and infers the copy number of the repeats. And this is from from array data, the data that you'd find in the GDC right now. And there's two kind of tied together parts of this. So there's the first part uses DNA copy. It's an R package to perform circular binary segmentation analysis on the normalized array data. So it takes the noisy intensity measurements and then transforms them into the chromosomal regions of equal copy number. And then it segments it into genomic region with the estimated copy number. So a newer part of this pipeline we've got is this copy number estimation. So if you go on the GDC portal, you'll be able to download these files. And it is numeric focal level CNV values generated using GISTIC. So these are gene level copy number estimations. And then the next page, again, shows you some examples of what those files will look like. So on the left is the first file. It gives you the chromosomal position as well as the calculated segment mean. And then the second file, that GISTIC file that I was talking about, the copy number estimate, will give a list of the genes. And then if this is on a project level, it will show you whether genes have a specific copy number loss or gain. So a 0 is neutral. Genes with less than 0 are considered a copy number loss. And then genes that are greater than 0 in this matrix on the right are considered having a copy number gain. So that's what you can use it for. And then again, a lot of this data is visualized on the GDC portal. So you can see specifically for example, each gene, you can see whether basically the quantity of cases that have a gain or a loss in copy number. So lastly, the methylation liftover pipeline. So what we've got is a level of methylation known as CpG sites, and then we calculate beta values. So we take the array intensities from aluminum methylation arrays, and we remap them to HT-38, and then associate transcripts from GenCode version 22, and then calculate the CpG island and site distance from each feature. And then the beta values that we're using are inherited from the existing TCGA level 3 DNA methylation data. I think, oh, I didn't put an example of what those files look like. I think they're pretty large. So the next thing I want to show, so those are all the pipelines that contain data within the GDC right now. I just want to show you where you can find that on the GDC. And then just do a little brief demo there. So I'm going to escape. I'm going to try, I'm having a little trouble with my computer. I'll stop sharing and I'll try it again. All right, I'll try and share my screen. Let's see if this works. Okay, yay, that's good. Okay. If you haven't been to the GDC portal, if you're new to the GDC, this is what it looks like. You go to portal.gdc.cancer.gov, and then this main page will show you all the different projects, primary sites, cases. Just gives you an overview of the data that we have in the GDC. So for some of the data that we were talking about from the pipelines, if you click on the projects page, you will see a listing of all the projects that we have in the GDC, and then a listing of the available data files that you can expect. So sequencing reads, transcriptome profiling, nucleotide variation. This just gives you a high-level overview of how many cases you would expect to have files for that, as well as the number of files that you would expect. So the main place that I recommend people go if they want to download, for example, some of the gene expression data or the sequencing BAM files, as long as you have controlled access, you'd go to repository, and then that loads a new page here. And then on the left, you see the files and cases filters, and you can start to see some of those data that I was mentioning before. And you can filter by different faceted search options. So we have data category, data type, and experimental strategy. So for example, if you're interested in RNA-Seq, you go to experimental strategy and say, I'm interested in RNA-Seq data. And then that filters the results, and this gets updated. And then you can see the different workflows that I was talking about that corresponded to that original image of the data model. And you can see all the different pipelines that were used to create all those as a filter. So for example, if you wanted the raw counts for a number of cases, you would just click on that. And again, it just filters all the results for you. It's kind of nice. And then, so this is for every single project in the GDC. Let's say you're just interested in a specific group of cases or a specific primary site. You would click on cases. And then, for example, if you're interested in TCGA, you'd click TCGA, and then a project that you might be interested in. So I'll just do LUAD. And then finally, on the right, you can see we've significantly narrowed down the number of files we have, 594 HT-Seq count files. What you could do is use that tool that I was describing earlier, or add them to your cart. This only does 20, I'll just do 20 for right now. And then go to the cart and then click on download cart. And what that does is it takes all those text files, zips them up, and then downloads them in the browser. Alternatively, if you wanted to download something like a BAM file, which are huge, gigantic, that's not something you would want to do on the web browser to download multiple terabytes of data. So we have a separate tool called the GDC client that lets you download BAM files. So what you do in that case is, let's say I'm interested in the same group of cases. Instead on the files, I will reset these. Oh, actually, maybe I'm interested in the BAM files for RNA-Seq, and I want to do my own analysis. So you'd come down here and choose a different data format. So instead of the derived files, you want the BAM files, and that lists all the BAM files here. As you can see, it's quite large. So you'd add them to your cart, come back here. Instead of downloading them, you would download a manifest file containing a list of the UUIDs and then pass that to the GDC client. We have another, if you look at our doc site and our webinar, we have some instructions on how to use that. So the other question I frequently get is how do you actually associate the metadata from those files to the actual themselves? If you stay on this data page here, these buttons here will help you a lot in determining which file will correspond to which case or the particular clinical metadata associated with it, with each. So if you download a sample sheet, it will have that association for you. So the last thing I wanted to touch on here was our documentation site. I did speak a lot about that, and I'll show you where to find that. So if you go to GDC Apps, and then you click on Documentation, and it loads, here we go. So here are all the different applications we've got for the GDC. If you click on Data, you'll see different options for the different pipelines that I had described. So DNA-Seq, for example, if I'm interested in that, I click on here. And then this essentially is a more verbose version of the slides that I presented that talks about the different processing steps, some of the familiar images that you've seen, and then the different commands that we use to process a lot of these. So if you wanted to replicate it, you could if you wanted to. So that's DNA-Seq. And then the other important thing I wanted to show, we get a lot of questions about that, is the MAF files and VCF files. It's in that same section. If you click on it, you'll very clearly see, like I was describing, all the column names and then descriptions of what each of those are for the MAF files themselves, which is super helpful to people. So I am going to go back to the slides. Where to begin? Okay, so the last slide that I had here was, I talked about some of the pipelines that have existing data in the GDC, but we're always developing new pipelines and improving on the work we have for those. So this slide represents the pipelines that have processed data. Those are the ones in purple. And then the blue ones represent these new pipelines that we have and planning to process and are processing data for. So I will just briefly walk through some of these. So as you can see, the purple ones are, the ones we talked about before, but the blue ones, we have Caveman, which is a new algorithm that will detect somatic mutations based on tumor-normal data, BRASS for finding structural variants, a newer version of Pindel, ASCAT, NGS, which will include copy number variation data for whole genome sequencing information, and then GATK4 pure CN, which is another way of calculating single nucleotides. And then on the top for RNA, we have ARIBA and STAR Fusion, which are algorithms to detect gene fusions. And then the copy number variation data are these last blue ones that we've got here. And I think that's pretty much it that I had for the slides. I will point out that we've got some reference files. I put these on the last slides for the previous webinars. If you want to know more about the website documentation, follow us on Twitter. We've got other, we have our support email. If you have really specific bioinformatics questions about how we process something, what commands we used, what versions of files, please reach out to us, and I will direct the questions to Zenu and his team. But I think that's pretty much it for the presentation. Again, I have my colleague Zenu here. He's the manager of the bioinformatics team. If you have any specific questions, please let us know.
Hi, Colin. Hi, Colin. There's a couple of questions that we did get in the chat. The first is, at what point does the tumor-only variant calling pipeline differ from the tumor normal pipeline? So can you talk a little bit about the tumor-only variant calling pipeline?
Yeah, so this is Jenny. Yeah, the tumor-only pipeline have a unique requirement because the normal pair is not there. And so the issue here is that because there are existing germline variants, we don't want have the germline variants leaked into the somatic host. The challenge here is how to remove that. So we actually, the new pipeline attacked this in two scenarios. One is we actually updated our MUTAC2 from the JTK3 that was for the tumor normal paired pipeline into JTK4. That provides better filtering and better calling quality for the tumor only. And also we plug in another tool called PureCN. You can see that in Colin's slide. And this particular tool actually trying to categorize the somatic status by computing some of the metrics such as the tumor purity, heterozygosity, and copy number changes, and try to predict when you see a variant that's somatic or germline, and it will label the ones with probability that allow for better filtering. So those are the main changes.
Thank you, Zanu. There's a question about sharing the deck or post it somewhere. So what we do is, after the video presentations, we then create the transcript of the video and make it 508 compliant. And we do put it on our, if you go to our GDC support page on the website, that's where you'll see the prior, webinars. And once we get it posted, it will be there so that you can access it. And it's if you go to gdc.cancer.gov, at the top, there's a support link, and that's where you would hit. And then you could see there is a menu item for the webinars that you can select. Another question, is there any possibility to download isoform quantification for coding genes like microRNA isoforms?
Are you talking about the microRNA or the mRNA?
It says microRNA.
Yeah, so we have two microRNA quantification files generated out of one input. One is the microRNA gene expression, the other is the isoform expression. And Colin, yeah, so Colin is pointing that out. So the isoform expression quantification, that's the isoform expression if you want to use that.
We just had a clarification from the person asking. It's mRNA and not microRNA. So,
oh, the mRNA, not currently. All the quantification is based on the gene level that HT-Seq and STAR both providing. We are thinking about adding another pipeline that can quantify for isoform expression. But if you are interested in doing it yourself, my suggestion is using the RCM tool for particular input. So we have different type of RNA-Seq aligned band files. For RCM, that's can only take the, we call it transcriptome aligned band. If you use that, you can do the quantification yourself. But we don't have it right now. We will consider implement that.
Okay, and another question, Do you support multiple tumors per patient ID, such as heterogeneity studies or tumor met pairs?
Yes. So Colin mentioned the adequate level maps. So the variant callings that we have is for every tumor adequates we have, we would find a normal pair, and then we will do variant calling. So the number of variant callings depend on the tumor adequate. If your patient has 3 or 5 different tumor adequate with DNA-Seq bands, all of those will be calling BCF, and eventually we will generate the same number of adequate level maps.
Okay, another question is, how are mutations aggregated from different software?
Oh, that's a hard question, actually. So, as Colin mentioned, we have multiple variant callers, somatic variant callers, and as they have their own unique calling set and also have some common set, the idea of aggregation, actually we first take every calling set and only looking for what this caller think is good mutation. So if there are future columns saying it's good, bad quality or genome leaking, we won't take it. So for every caller, we will keep a list of good variants out of it, and then we apply a rule called 2 callers criteria. So we only allow mutations to get into our public access maps when it's being called by at least 2 or more callers in this perspective. We do have some other GDC implemented specific germline variants filtering, such as we have to looking for the normal depths, make sure the normal pair have enough depth in the variations, and also require the variant to be in the exon regions and also within the packet capture kit region. There are some other small filtering steps we did, but the 2 caller criteria is the main thing we did for the multiple caller aggregation.
Okay, there's another question of any idea how whole exome samples with less than 100x MTC perform in terms of variant calling?
I haven't checked that specifically. I don't think that's a big issue because whole genome data normally have a less coverage. So whole exome less than 100 coverage, you can expect some of those may be missed because of coverage, but in general, I think that's fine for most of the variant detections.
Okay, we have another question. What is the copy number resolution with the 2 software mentioned? And are there any limits of detection specified for the data sets?
Oh, so 2 of the software from Colin's slides, one is, well, actually both are ASCAD. One is the ASCAD for the SNP platform. The other is ASCAD for the whole genome platform. We haven't looked at carefully at that. For our read platform, then normally it's very straightforward, depending on the inflows between the probes, and that's at the resolution of the probe level. But there are going the circuit battery CBS algorithm or some other segmentation algorithm. I think SCAD probably is a different algorithm, but that's the resolution. The whole genome, I won't say it's really as accurate as base level. That's really based on the coverage, but just less than megabytes level. I would assume it's in the KB region, but I'm not really sure. It's actually best to look at the publication as a single tool called ASCAD. That's, I think, easier to check with them.
Okay, a couple of other questions. What is the typical size of the samples being taken through the variant pipelines? Any impact to pipeline parameters or output if the samples are only 30 to 50?
Could you repeat the first half of the question again?
What is the typical size of the samples being taken through the variant pipeline? Are there any impacts to the pipeline parameters if there's only like 30 to 50 samples?
Thanks. So the typical size, we have very different sizes. I will make TCGA as an example. For whole exome, the typical size of tumor is about 150x, and normal is normally less. For the whole genome data from TCGA, the tumors are normally 40, 60x, and normals could be 30 to 40x. And the less the coverage is, the not ideal the result you'll get, but we do have some communication with the author of these individual tools, and they are very different. For the whole genome tool, we have and is in production. That's from Sanger. What I heard is even you get to as low as 15x. The data, although it's not ideal for checking individual mutations, but for mutation signature purpose, that's still very useful. So that's a general idea. But also, the sensitivity is dependent on tumor purity. So make sure if you have a less pure, more heterogeneous tumor sample, it's better to have a higher depth. If the tumor is 100% pure or very high purity, and a lower depth normally is not a problem.
Okay, we have another question. Do you have a structural variant pipeline for whole genome sequencing?
Yes, so that pipeline called the BRASS. It's a structural variant pipeline we just developed. That's also a tool from Sanger. And you can expect two outputs from it. One is the DCF file, and the other is the BET file output. This data haven't been released yet. We just started the production stage, but you can expect that probably some project will have this data by the end of this year, I guess.
Okay, there's another question on how does the GDC ensure quality of the data generated using GDC pipelines?
That's a good question. So there are multiple steps of QC in the pipeline. Actually started from tool selection, and then initially we also do a lot of benchmark. And for every tool pipeline we developed, we do a small production trying to compare with existing data from other projects or from other similar tools to ensure the quality of the tool. And during our pipeline development, there are multiple QC steps inserted directly in the pipeline, and we do collect a lot of metrics also after pipeline production and to make sure everything makes sense. And at the final step, actually, after data is produced, often we actually give the data to the submitter, those owner of the project, and they will help us do a final review of the data quality. And that's also a very important step before we actually release the data to public.
Thank you, Zhenyu. And I want to point out too that there was a recent publication that was submitted. It's available on the GDC website under publications. There was an analysis working group called the GDC Quality Control that was composed of several members from TCGA that did a comparative analysis between the TCGA HD19 version and what was produced by the GDC. And that, they found very high concordance. So that's another way we did quality. There's another question that just came in. How often do you update gene annotation? For example, you are using GenCode version 22?
Not often, and we don't have a plan yet. The reason of that is it's heavily integrated in a lot of our data sets, and changing the gene annotation is actually very hard. It's not only for the DNA-Seq variants, which is relatively easier. We can just make changes in the last step of annotation and in the math, but bearing in mind that RNA-Seq alignment itself actually take the gene model into consideration. So updating a gene model that requires reproduction of all the RNA-Seq from the beginning to the end. And so methylation data also have all these heavily incorporating their data set. So definitely we'll consider if there is a compelling reason to do that, but it's not in the current planning yet.
There's a question on how is your pipeline different from MC3?
Yeah, so thanks for this question. So MC3 refers to one of the effort a group of researchers from the consortium that's composed of a lot of the original TCGA analysis one groups, and they use 7 different colors for whole exome data reprocessing, variant calling. And we have close communications with them, and a lot of the current strategy of editorial level math was taken from them. And among those 7 colors, and we actually share, I would say, 4.5 of those. So GDC has 5 colors that Colin mentioned. 4 of those are the same. The half one, what I mean is they use Mutec 1, and we actually use Mutec version 2. But they do also have 2 additional colors. One is indel locator for finding indels, and the other is a color called Radia. That has the option not only use tumor normal pairs from DNA-Seq, but also can take a third RNA-Seq band. So that's we have a different set of colors, but a lot of the downstream processing, including filtering and aggregation, are very similar.
We have a question on how can you comment on using HD19 versus HD38?
Yes. So definitely suggest you use HD38, and it's not only because the coverage of the genome is higher, but also it's more accurate. It contains a lot of sequence previously cannot be located, or previously had heterogeneous bases on those locations. The challenge we actually first met is that was first production about 3 to 4 years ago, and there are a lot of the data resources and tools that are not compatible with the new reference genome. But nowadays, since the HD38 has been released for almost 6 years, I think most of the modern tools have been updated to be used with this new reference genome, and it's definitely better. And you can see most of the other big genomic centers also move towards using this new reference genome instead of staying with the old one.
Okay, and another question, are there variant callers or other algorithms in general that we've tested but chosen not to implement in the GDC?
Yes, we actually benchmark a couple and also read the literatures about different things. And I won't list a particular one, but I can tell what we are actually looking for. So one is the accuracy we compare with others, but a big part is for the stability. So a lot of the bioinformatics schools, they often being developed by graduate students, and they work fine with a couple hundred of files. And when we do a GDC level production, that means hundreds of thousands of in that level, sometimes some tools, although the accuracy is okay, they will have a big failure rate. And sometimes we work with the author to get this updated, but that's not always the case. We can get it done. So that's another big consideration when we pick tools.
Okay, and that's actually the end of the questions. So I think we can close this webinar. Thank you, Colin and Zhenyu from the University of Chicago. Thanks, participants, for calling in and joining us. Have a good day, everybody.
Thank you again.
Thank you.